So here we have these are the things we're most interested in following up. This is just a reiteration of the MMP7 data. In this case, we've normalized the apoptosis to the cell number, and you can see more clearly the level of impact on apoptosis that you're having by knocking down the MMP7 in the RAS mutant cell lines, but not in the wild-type cell lines. So a little bit about MMP7. It's one of a large family of matrix metalloproteases. There's a lot of evidence going back quite a long way that they're important in cancer, and particularly there was a lot of focus on them in tumor progression, and they're upregulated in many human cancers associated with poor survival. This led to the development of a lot of inhibitors of MMPs 10 or more years ago, and putting those into trials. There were a lot of trials conducted on these, and I think it's fair to say they failed in a fairly spectacular manner, largely, but not exclusively, down to the severity of the side effects, in particular problems with bone remodeling and acute pain due to inhibition of that. MMP7 itself is unusual in being expressed in the epithelial cells rather than the stroma cells, which is where most MMPs are expressed, and hence, I guess, why it could show up in our screen. There are a number of ways in which this could be acting. There are, taken from the literature, a few different ways in which MMP7 mechanistically could act to promote survival. And just say that actually one of these we've looked into in some detail, which is the plugging into the Wnt signaling pathway. And this does indeed appear to be the way in which this is having the effect in the colon cancer cell lines, although we have not really been able to provide convincing evidence of that in the lung cancer cell lines as yet. There may be a different mechanism in the lung cancer cell lines. But basically what it does is it cleaves E-cadherin and releases beta-catenin into the cells where it promotes Wnt transcriptional response. So maybe targeting MMP7 could be helpful in RAS mutant cancers, and they're extremely druggable genes. These just show one of the drugs that was in trials originally. It does show some activity in our panel, but this is a broad specificity. They were all broad specificity drugs that went into the trials, and it may be that inhibiting other metalloproteases is a problem. We've done, like everyone doing these things, we've done various sorts of network analysis. I think I need to skip through this quite quickly now, but basically that showed an importance of proteasome function, and this was something that Steve Elledge's lab, who also have done a related sort of screen, have seen evidence of this as well. And if we use proteasome inhibitors, we can see they selectively, so here anything above 1 is selectively killing the mutant RAS mutant cells versus the wild type cells. At the same time, we also did a library screen of drugs of known function or suspected mechanisms of function, and this came up with a few interesting things, most notably drugs targeting topoisomerase function, and this was actually something that had been picked up a few years ago by Brent Stockwell. And so things like camptothecin, topotecan, etoposide are really quite selective for RAS mutant cell lines, or to somewhat selective. So just a word here about this difference between synthetic lethality and oncogene addiction. We think that there are two distinct ways in which these are operating. One is the synthetic lethal effects are short-term clashes of positive and negative signals that the cells experience as a result of the oncogene expression and whatever therapy you're throwing at it. And for example, topoisomerase inhibitors or the CDC6 siRNA that's targeting a DNA replication licensing factor seem to work in this way. They will kill cells where there's an active RAS signal. We've looked at this by reintroducing a rapidly inducible RAS into our cells where the RAS had been removed, the RAS allele had been removed, and those cells don't become addicted to the RAS again in vitro. We think that actually addiction to RAS requires selection within the tumor environment and is not something that is truly achieved in vitro. And you switch RAS on, the cells are sensitive to these things. If you switch it off again, the cells lose the sensitivity. The oncogene addiction is rather different. The cells, the RAS mutant cells are addicted to the oncogenes, and you can't set that up in vitro. There's also this addiction to non-oncogene pathways, so these are really the stress response pathways, and certainly the proteasome inhibitors plug into that. So a tumor cell with activated RAS will show this. If you remove the cells where we've removed the RAS, then we reintroduce it, they don't show any sign of this. They don't show, just having activated RAS doesn't make you sensitive to proteasome inhibitors. So it's something that reflects the evolution of the tumor. And we think that there's some mileage, at least, in targeting combinations of these mechanisms to get selective, improving the selectivity of the mutant versus the wild type. And I won't go through this in detail, but just to show the sort of the type of improvements you can get. So here we're using low doses of topoisomerase inhibitors and proteasome inhibitors. And if you get the combinations right, you can actually start increasing that selectivity quite markedly. And there's an interesting effect that's shown in there of actually scheduling that if you treat with the topoisomerase inhibitors first and then the proteasome inhibitors, you get a remarkably better effect than if you do it the other way around or the two together. The explanation, the mechanism for that is still not completely clear. And then also finally, just to show that if you put in the MMP7 inhibition at the same time using the MMP7 siRNAs, you can also get big improvements. So here you have the effect of the MMP7 siRNA by itself and then a sublethal dose of bortezomib, and then putting the two together, you really improve the selectivity, and this is the same done with topotecan. So there's mileage in trying to combine these things together to get improved selectivity for the RAS mutant genotype. So just to summarize that, we found these 50 or so hits that were selective in the colon cancer system, the HCT system, and extended them to a larger panel. And we found these dependencies on proteasome function, topoisomerase activity, and evidence of non-oncogene addiction being the mechanism for proteasome function and MMP7 function dependence, synthetic lethality being the mechanism for the topoisomerase or the CDC6 dependence. And combining them together can have improved selective effects. So this is a figure I borrowed from the Steve Elledge review last year, and it just shows that he's extended the idea of how many pathways are required in a cancer to include a lot of stress management pathways. And we would think that the RAS oncogene addiction is probably around this dependence on pro-survival signals, which the oncogenes are actually generating. But these stress pathways explain some of the other phenotypes. The RAS synthetic lethality is probably, at least as far as the DNA damaging agents is concerned, is really that the cells are already tolerating DNA damage and can't take more of it. And the proteotoxic stress is also something that reflects this non-oncogene addiction to proteasome function that they require. And if we can target these together, we may have improved outcomes. Many of these are actually targetable by drugs that already exist, so I think there are interesting possibilities in combining them together in the clinic, which largely has not been done. Okay, I'll just reiterate the thanks to the people who did this work, particularly Tony Ramjan, Surbi Gupta for the PI3 kinase mice, and the Dave Hancock, Michael Steckel, Miriam Molina for the last bit I talked about. For our screening work, our bioinformaticists, Gavin Kelly and Phil East, have really been essential. Thank you very much.